All right. I'm a PhD student in Christian Schletterer's lab, and I'm going to talk to you about low concordance of short-term and long-term selection responses in experimental Drosophila populations. So this talk is all going to be about experimental evolution. And experimental evolution is a take on the question, how predictable are the outcomes if I replay life's tape? And while this question has asked a couple of years back more on the phenotypic level, now with next generation sequencing, we have the possibility to look at this on the genotypic level. Okay, so how can we study adaptation in the lab? We call this evolve and resequence. So that's a combination of experimental evolution and next generation sequencing, which allows you to study adaptation under controlled conditions in a replicated manner. And the goal that you have is to identify contributing pathways, genes, or even SNPs. This approach is quite popular by now. So here are some papers in case you want to learn more about it. And the studies that are published right now come in all terms of flavors. So we have different selection regimes that are investigated. So just listed a couple here, like virus infection, host pathogen interaction, thermal adaptation. This is what we do a lot in Christian's lab, body weight, but they also vary in the sensor size that is used in these studies, the replication level, the history of the ancestral populations, whether you apply artificial or natural selection, and the experimental duration, so the number of generations. And this is what I want to talk to you about today, because the empirical studies that are out there, they vary considerably in the experimental duration. So for Drosophila, we have studies that go on for less than 20 generations, but on the other hand, we have studies that are maintained for hundreds of generations. And this is kind of interesting because simulations have suggested that with short experiments, you have lower power to find your selected alleles. I brought you one example. So this is a simulation study conducted by Robert Kofler and Christian Schletterer a couple of years back. So you have two panels here. So panel A is for strongly selected alleles and B is for weaker selected alleles. And these are ROC curves. So like Kelly said, we have on the y-axis, we have the true positive rate. On the x-axis, you have the false positive rate. And if you have a really good setup, what you would like to have is that your curve is hitting the top left corner. And the different colors that you see are different experimental durations. So black would be the experiment with the most number of generations. And what you can see is that you have more power with a longer experiment. This has not been only shown in this study, but in a couple, so this is nothing new. However, what was missing so far is the empirical investigation of the differences between short-term and long-term genomic signatures. And this is simply the case because there are not a lot of studies out there where you have all the intermediate time points sequenced. But I'm happy to tell you that now we have, and I will present to you results that are based on an experimental Drosophila simulans population that originated from Florida. And they are exposed to a new temperature regime. So this is the study I talked about thermal adaptation. And NEDA actually showed that the response is highly polygenic. So you have a lot of loci that are contributing to adaptation. And what is really nice about this study is that it's highly replicated. And we don't only have 2 time points, but we have 7 sequence time points. We use pool-seq for the sequencing. So what you have there is a really robust allele frequency estimate. And based on the allele frequency estimates and their changes, you can determine candidate SNPs that are most likely contributing to adaptation. And the properties of the study not only allow me to compare the most distant time points, which is shown by this blue arrow, but I can also look at early selection signatures or do all kind of pairwise comparisons. Okay, and this is what I did, and I did this on 3 levels. So the first level, what you can do is you can take your outlier SNPs of each evolved generation and simply compare the similarity of the sets. However, as most of you guys know, especially neighboring SNPs are most likely not independent from each other. So it would be a good idea to kind of go one step further and try to estimate the linkage somehow, such that you don't run in a lot of false positive signatures. And one way to do this are candidate windows. So what I did is I split the genome into windows of a certain amount of SNPs. And then I used simulations to determine which ones have an enrichment for candidates. And then similar to the level before, you can look at the similarity of your candidate windows that time. However, this data set has additional information about the linkage structure because NEDA clustered the allele frequency trajectories of the candidate SNPs, and this results in haplotype blocks. So you have an idea about the linkage structure in the evolved populations. And what you can ask is, if you look at the marker SNPs of these haplotype blocks, how many of them can I find actually before? Amongst my outlier SNPs. And this is something that I called haplotype block discovery rate. So I brought a toy example here. So I don't know, I hope you can see my mouse. So we have 5 marker SNPs for this haplotype block in gray. And then out of these 5 marker SNPs, you find 3 already in generation 50. So your HADR would be 3 out of 5. Good. So just a recap, we have these 3 levels, and now we're going to look at the results. So the first thing is that subsequent time points are more similar for advanced generations. Okay, so I'm going to show you the summary of this statement. So what you see in A is a matrix of all the pairwise comparisons of the evolved time points. And in the top triangle, you have the results for the candidate windows. And in the bottom triangle, you have candidate SNPs. And the single tiles are colored and labeled by the Shakar index. And the Shakar index is a similarity measure. So if it's 0, this means you have no overlap whatsoever. If it's 1, this means the sets are actually identical. And what you can see for both analysis levels is that early generations, early neighboring generations have a lower Shakar index than more evolved time points. And you see a similar pattern if you look at this HADR statistic. So only later on, this HADR statistic actually gets higher values, like on average above 50%. Okay, and this suggests that a more reliable identification of selection targets would strongly benefit if you let your experiment run longer. However, I would like to point out that this is still an empirical study, and although it's done really, really carefully, we don't know the true targets of selection, and this assumes constant selection over time. However, I have performed simulations mimicking this setup, and with simulations you have a gold standard, so I really know which ones are the target of selection, and I observe similar patterns than I just have shown you. I just have to skip this because of time restrictions. Okay, so the next finding is that only few selection targets are shared across all generations. So what do you mean by that? Here you have a plot where you have on the y-axis, you have the fraction of rediscovered candidate SNPs of generation 60. And on the x-axis, you have the intermediate generations. And the color of the bars tells you actually which level are we looking at. So if it's the candidate SNPs or the candidate windows. And a value of 0.2 would, for example, mean that 20% of the candidate SNPs in generation 60 are statistical outliers all the way back to generation 30. And what is really easy to see here is that the window bars are always higher than the SNP bars. And this suggests that window analysis show more consistency than SNP-based analysis, which is probably not too surprising. However, we recommend that if you compare ENR studies, for example, you should do this on a window level or selected haplotype blocks if you have, because this will help you to avoid false negatives due to the high stochasticity of SNP-based analysis. And Neda and Christian wrote a paper in Molecol where they actually discussed this. So feel free to look it up if you want to learn more about that. And again, I confirmed the I used simulations and I could confirm my empirical findings. So last but not least, selection signatures detected early in the experiment are not representative of the underlying adaptive architecture. So just as a short recap, this is based on this third analysis level, where I look at the rediscovery rate of these haplotype blocks. And as I have 5 intermediate time points, I can do this 5 times, get like a time resolved statistic, and I can perform hierarchical clustering to group them by the similarity. And if I do so, I actually find a cluster of 10 blocks that have a really high HADR, which would be the yellowish color, very early on in the experiment. So this would be a hint that you would find these selection signatures very early on. And I got kind of interested, so how do they differ? And I'm gonna start with how they don't differ. So you don't see any significant difference to other haplotype blocks in terms of starting allele frequency, the length, the recombination rate, absolute selection coefficients for the first 20 and the whole 60 generations, the number of rising replicates in generation 60. However, how they do differ is that they are stronger selected at earlier time points. So how can you determine that? You can calculate the selection coefficients for the first 20 and the whole 60 generations, and then you build a ratio. And what you see is that these early detectable haplotype blocks, which are the blue ones, they have an enhanced ratio. And the different panels are actually different allele frequency change thresholds for the haplotype blocks in one replicate to be considered for the analysis. That's not so important here. The main point is this pattern is robust to the filtering that I apply. And consistent with this finding, what we see is that EDHAS also rise in, it is early detectable haplotype blocks, rise in more replicates in generation 20. And this is something we link to the winner's curse. So what does this mean? Loci where stochastic effects increase in frequency in multiple replicates can enhance the contribution by selection. And this makes them to be more likely to be detected if you do a genome-wide scan. And again, simulations confirmed empirical findings, but this time I'm going to show you. So I simulated unlinked loci that have the very same properties in terms of starting frequency, selection coefficient, dominance factor, and then I applied our pipeline to scan for candidate SNPs. And what I found is that although they have...
Sorry to interrupt, Anna, you have 1 minute left of your presentation time.
Thank you, Neda. Although they have the very same properties, they rise in more replicates. So drift acts synergistically with selection and makes them detectable. And what this means is that what you find after 20 generations of adaptation in this experiment is actually more parallel. And this could result in a biased picture of the underlying genetic architecture, because simply by the fact that drift and selection are working in the same direction, it is more likely to detect parallel than replicate-specific selection signatures. And for future work, what would be cool to do is to scan short-term experiments for an enrichment of these parallel selection signatures. And with that, I would like to thank you for your attention, and my lab for a lot of support, Christian for supervision, and Neda, Claire, and Shankai, who gave me really nice feedback on previous version of this paper. So you can find the work I just presented in Molecule, or you simply scan the QR code here. And thank you. I'm happy to take questions now.